And as Peter pointed out, I am not a medical doctor. Thankfully, I do not have to have the answers like these very learned speakers before me. I get to have the questions, which is much easier and for me, much more fun. And one of the major questions that I'd like to talk about today is, how do we discover new drugs? What on earth do we do? Thousands and thousands and thousands of genes are all running amok, having a great time, and suddenly something goes wrong. How on earth would we find out what's going wrong and then how to make it go right? And one of the major reasons, one of the major avenues we have for research is that sometimes nature gives us a little hint, which we are very grateful for, with some natural experiments. Oops, there you go. So, some of nature's little experiments. And you might look at Robert Wadlow or Sandy Allen and think, well, maybe we might learn something about growth from this situation. And in fact, we did. And just as an aside, he was 22. Yes, extraordinary. And we learned about pituitary hormone and how when you get a tumor in your pituitary, that increases the certain hormone, growth hormone and others, that you get growth. A perfect little experiment done for us, and we can try and take advantage of that. This is a Belgian blue. Look at that for barbecue heaven. And so from this, a spontaneous breed of cow and bull, we learned about how muscles grow. We learned about myostatin. Now, and so he grows so muscular because he doesn't make myostatin. His myostatin gene is broken. So as John mentioned before, an antibody to myostatin, something that will bind it and make it not work, is now a treatment for people who can't grow enough muscle. A perfect example. And I've actually worked with the mouse version of these guys. We call them Mighty Mouse. They're enormous. I've worked a lot with this guy, so-called Obi mouse. No guesses for why he's called Obi. A spontaneous mutant found in a colony one day in the US. And we learned about a hormone called leptin, which dominates how your body survives in terms of starvation and calorie excess. Gorgeous little things, little fat, chubby. Anyway, aside. So, the question becomes, what about bone? Well, we learned something about bone from all of these. We learned about how bone length is controlled. We learned about how the weight of bones is controlled. They have very big bones, as you might imagine. And he, believe it or not, has very small bones. So we learned something about bones from all of those, but hardly an appropriate treatment platform, perhaps. I mean, some gentlemen might quite like that, but most wouldn't like this, and I think that'd be a bit tricky. And so we do look for situations where it is primarily a bone effect. And we do know some of those diseases such as osteogenesis imperfecta, and there are a range of types of OI, and you can see the various impacts they have, from the quite mild to the absolutely extreme. And from that disease, we learned a lot about how collagen, which is the protein that bone is built on, how collagen affects the quality of bone. They get quite a lot of bone, but it's very chalky, and you can see that its ability to be an appropriate structure fails. And so there is one disease that has come along, and John mentioned this, and I'll tie in a lot with what John said, a Van Buchem disease. It's also sclerostosis. This is an X-ray of a normal skull. I want you to pay attention to how lovely and thin that skull is. That is the skull from someone with Van Buchem disease. Look at that. Staggering. And the other thing is the jaw. You see the jaw? Massive. But the one thing about this disease, well, two things that are an advantage. One, it very particularly affects the skeleton, and two, the bone is normal. It's not like OI, it doesn't break. these guys fall, 2 floors out the house, nothing. Get hit by a car, they can't swim because they sink. They just have a lot more bone. All right? So nature has done a little experiment, and for us, that produces a scenario that we might like to mimic. Maybe not this much, but certainly the ability to grow more bone and more good quality bone. That is exactly what we've been after. As John mentioned, a lot of our drugs were based on stopping bone loss, and that has historical reasons. During menopause, there's a lot of bone loss. So what do you look for? You look for things that stop bone loss, and that's fine. But bone is absolutely unique. It is the only tissue that will heal without a scar. It is the only tissue that carefully and precisely replaces itself, just like John said. So if you can get bone to form, it's the one thing you can make young again. Yeah, your skeleton can go back in time if you can put more on and stop taking more off. It is unique in that. And so this scenario offers us a potential platform to try and mimic what's going on. And so this is the classic picture of sclerosis and Van Buchen's disease. One skull, that's an affected person, more than 3 times as heavy. Their bones are big. And we want to try and find out about how that happens. And so as John mentioned, this person here has a loss of the protein sclerostin. So their gene for sclerostin, that normally is the template that protein's made on, is broken. So they don't make this signal. And that is all. That is the only thing that's changed. And the great thing about sclerostin is it is really expressed in bone and almost nowhere else. So you don't suddenly get big elephant ears and grow 3 legs. It's just in your bone. So it gives us a sense of control that as a therapeutic you might value. And so sclerostin is made by the cells that are in bone. John mentioned the osteoclasts that remove bone, the osteoblasts that make bone, and that's all on the surface, so that's up here. These guys are called the osteocytes, and they're in bone. That's the surface of bone, that's the surface of bone, and they're in. And they're all the way through it. See, that they look like little nerve cells, right? That would be where the cell is, and here's all its little legs. And they all stick out, so his stick out, and they connect with his. And his connect with his, and his. And so there is a little network all the way through the bone, right? And that's filled with water. And when I jump, my bones squish. And when they squish, they get smaller, and that shoots fluid through those little holes. And he notices. So he gets squished. And when he feels that squish, that is loading. And that's his job, right? His job is to go, yep, we're loaded, it's all good. And if we're loaded too much, let's make some more. And if we're not loaded enough, let's not worry about it. Okay, so these guys sense mechanical loading. And that is one of bone's jobs. It is very rare in that. It is a structural member. It has to be somewhere and it has to do something, or you're a jellyfish. Okay? And these guys stop that. And they stop that by reducing the amount of sclerostin they make. So when they get squished a lot, stop it with the sclerostin and up go the osteoblasts and they make more bone. And as you can see from the individual with Van Buchen's disease, they make a lot when they want to. So mechanical loading means less sclerostin, and less sclerostin means more bone formation. So that is a real opportunity, because less sclerostin doesn't really mean anything other than more bone formation. No extra ears, nothing. All right? And so we can make mice. So we can make a mouse that is a copy of the human scenario, so it just doesn't have that gene. The little minus means we cut that gene out. And this is a cross-section of his leg. That's the femur. The kneecap would be down here. This is normal, and this is one without any sclerostin. You can see the amount has almost gone solid. So it does recapitulate in a mouse that if you take sclerostin out, you get more bone. You can see this is a cross-section of the spine, same thing. Wild type makes more. You notice that they're not a crazy shape. That's very important too. It doesn't form it on the outside and you'll suddenly get it, right? So it is very controlled and it's very good bone. So it's a wonderful target. So the question is, can we block sclerostin? We did it genetically in Van Buchen's disease. Nature did it and we did it in the mice. Can we make a drug that does that too? Yes. They call them ridiculous names. Don't ask me. I won't even say it. But so this is an antibody. It's just, all it does is a protein that will link onto sclerostin and make it unavailable for anything else. It just, it clots sclerostin, if you like. And so this is an animal, a normal animal, and this is the bone when you treat it. And you can see the changes are enormous. And again, the bone doesn't suddenly get this big. It still looks normal. You're not going to suddenly look like a Cro-Magnon. And so this is what happens. This is actually the surface of this bone. So that's the bone marrow, and the black stuff is the actual bone tissue. And can you see the bright green lines on the front? That is bone formation. So we mark the calcium goes into the bone, and it glows bright green wherever it's making bone. And you can see that in the vehicle, there's some little green lines, and in the two sclerostin antibody treated, the lines are huge. So all we do, as John said, is we just ramp up the amount of bone formation because this guy thinks he suddenly weighs a ton, right? And he responds naturally. So bone loading is a natural process. He just thinks you're heavier. He doesn't think you're an elephant or a moose. He thinks you're a human that weighs more. So it responds very naturally and very appropriately by putting bone on where it thinks it should if you're heavy. So it's a very natural process. And John showed this slide before, but I'll repeat it because it's important. This red guy down the bottom here is the best thing we've got. There are two of these drugs. This is one of them, and this is the other one that hopefully will be out very soon. This red one, an injection every day, costs a lot, difficult. This one potentially was a 6 monthly. So you can see it dominates the scene, right? So it is a fabulous, we're all fingers crossed, it's hopefully gets FDA approval in the US very soon. And for mine, it will change the landscape if we can get people to take it. Now, this is a bit of work that Peter Croucher and my wife actually have been doing, not only in terms of osteoporosis, but there are other scenarios where bone loss is severe and has catastrophic effects. And that certainly one of those is cancer-induced bone damage. Very common diseases such as prostate cancer or breast cancer love to live in bone. You don't die of your primary tumor anymore, you die of your secondary or metastatic disease, and that is very often in bone. They are very aggressive when they get there, they're very painful, and they're very difficult to treat. Pathological fractures, it's very difficult. So can we try and support the skeleton and give it enough time that we can treat the cancer while it's there? And this is an animal model. This is upside down, it's actually the tibia, it's your shin. So your shin, the sharp bone of your shin is right there, that's the front. That's the knee, and the femur I showed you before will be up here. No tumor, and they'll look all lovely and intact. Tumor. Not really much to say about that. Very, very aggressive, very destructive, very painful. And the last thing someone needs when they've got cancer is to have to worry about their bones as well. Enough. And so this is tumor with sclerostin antibody that they've applied, and you can see, look at the change. Look at how much more intact that guy looks than that. And so that is a fabulous potential to move from not just osteoporosis, which is a very common and severe problem, but onto very active and very aggressive scenarios such as bone cancer. So just to wrap up, the search for new therapies. So our genes tell us how we work. They are the instructions, and we obey. There's genes and there's environment. You can eat McDonald's all day if you like. I'd probably still stay skinny. Lucky me. Most people not, right? Your genes are how you respond to your environment. Sometimes when things go wrong, they also go right. And it is up to us to try and utilize them as much as possible. The exciting thing is that our current genetic information really is the tip of the iceberg. The ability for us to understand my genetics, your genetics, is skyrocketing. It took decades to do the first genome project. It takes a week. It'll take a day. And so we are cracking it with ever more speed and accuracy. And so that provides us with opportunities to discover new drugs. Is there any particular gene that might respond in any particular way, just like sclerostin, is a potential for us to use that to help people who have diseases such as osteoporosis. Thank you very much.